Hello and welcome to this rapid crash course for Prelims by Shikara Academy. So in this course, we'll be completing all the science and tech syllabus asked in various examinations such as UPSC Prelims, TSPSC Prelims, TSPSC Group 1, Group 2, APPSC and all the other State Public Service Commission examinations. So this is our second live session. It is our class number 2. And in the previous class, we have discussed a bit about the cell, about the genetics, what is a chromosome, what is DNA. We discussed about the sex chromosomes, we discussed about the ploidy, and today we'll be discussing about, let's start with genes. So we have seen that the DNA is a long sequence of nucleotides. We know that DNA is a long sequence of nucleotides and the condensely packed DNA, the more and more condensely, the maximum condensed DNA is the chromosome. But the chromosome is a larger entity and DNA is its constituent. But on DNA, the entire DNA that we have in the body, it is not, we will say it is not useful or that it is not functional. Okay. So the entire DNA is not functional. Only some part of the DNA is functional. So the functional sequences. So if a DNA is a very long chain, more than 3 billion nucleotides long, our human DNA is more than 3 billion nucleotides long. So any functional sequence of nucleotides on DNA is known as genes. So for example, if we take this sequence, this is an example sequence. In this example sequence, if this portion of or this sequence is coding for certain proteins, is coding for certain characters, then we say that it is functional. So it is a functional, if it is functional, it is gene. That is, it is coding for certain proteins. It is determining the characters, it is determining the traits in the individual. Moreover, 95% 95% of the DNA that is present is non-functional, or we say it is non-coding. So we call it the junk DNA. So 95% of the DNA is junk DNA, and only 5% of the DNA is making the different genes. So we have approximately 30,000 genes in humans. Approximately, the number keeps on revising, and our average gene is 4,000 nucleotides long. So we have 30,000 genes approximately, and approximately an average gene is around 4,000 nucleotides long. So if we take the analogy, if we make the analogy between, so we can take the nucleotides as a letter of the English alphabet. So the nucleotides that we have A, T, G, C, we can take them as alphabets, that is from A to Z. These are like the alphabets or the single entities. Then we can make the analogy of gene as a word, a meaningful word. And we can make an analogy of the DNA as a sentence. So gene is equivalent to word in the analogy. When if we are talking about the analogy, then nucleotide can be an alphabet and the entire sentence is the DNA. Now, while writing a sentence, we can have certain errors in. Okay, we can make certain mistakes. We can have certain spelling mistakes. So these spelling mistakes, in terms of biology, we can call them as mutations. So mutations are nothing but the sudden heritable change in the gene sequence that alters the gene function. So if we are given a sequence like this, and in this sequence, if we got at any point a heritable change, a sudden random heritable change. So this T gets replaced by C, for example, because of some random changes, because of some heritable change. But this C is getting heritable, that is, the DNA is replicating itself, and this is passing down through generations. And now it has altered. So now, if this gene was coding for something, suppose if this gene was coding for something X, then because of the mutation, either the gene can stop coding for that particular thing, or it can start coding something else. So we can say that the mutations in genes can lead to diseases. Can, please make sure that it is written can. It is not sure that it will 100% be making you sick or it will be 100% causing the disease. Now, mutations in the junk DNA. Mutations can happen both in genes as well as in junk DNA. So gene is the functional part of the DNA and junk DNA is the non-functional part of DNA. Now, if in the junk DNA, so if this, for example, if this is the junk DNA and this T gets replaced by a sudden random heritable change and the DNA has replicated itself. Now what can happen is if this was non-functional till this point of time, but now it can become functional and can start to code for certain things. So we can say that this non-functional or this junk DNA now has become a gene. So, and this formation of the new genes, so this giving rise of new genes, the new genes over the accumulation of the new genes or the formation of new genes ultimately can lead to the formation of new species, and that is the process of evolution. So, mutations in junk DNA they drive the evolution. Now, mutations can be of two types. So, mutations, we can have the point mutation that is on a single nucleotide. Also, we can have the frame shift mutations that is inserting or deletion of three nucleotides, at least three nucleotides, which is shifting the entire framework through which we are reading that particular DNA. Okay. So if this is now, if this at single nucleotide is changing, either it is getting replaced or it is getting deleted, we can say that, okay, the mutation has occurred at only a single point. We call it a point mutation. Now, if we insert if we insert three or more nucleotides in here, now we say that the framework of this or this entire sequence has shifted, and that is the frame shift mutation. Frame shift mutation can also occur when there is deletion. If we are deleting three or more nucleotides, then also we can get the frame shift mutation. Now we have seen in the previous class, in the last class, we have discussed about the gamete formation because of due to the process of meiosis. Okay, we know that the human cell in humans we have dizygotes, like are diploid. Human cells are diploid cells, that is, we have 46 chromosomes. But after we undergo the process of meiosis because of the gamete formation, so each gamete only gets 23 chromosomes. That is 22 autosomes, or that is the autosomes, and one sex chromosome. So a female gamete, that is the egg or the ovum. So if this is one egg or one ovum of the female, so it will have 22 autosomes and one sex chromosome. And in female, we know that the sex chromosome is the X chromosome. In case of male, the gamete formation it takes place, that is the sperm cells. The configuration is like 22 autosomes and one sex chromosome in the gametes. So we have 23 chromosomes in the gametes, and we say that the gametes, they are monoploid or they are haploid, and haploid of diploid is the monoploid. So we say that, okay, males, we have one X gamete and one Y gamete. So if the process of fertilization takes place, this process of fertilization or the fusion of the two gametes together. So the possibility here is that after the fertilization, we can get a zygote in which we have 44 autosomes. That is 22 autosomes and one sex chromosome is coming from female gamete, and 22 autosomes and one sex chromosome is coming from the male gamete. So we say that here we have 44 autosomes and two sex chromosomes. So total 46 chromosomes are there. So the zygote that is formed after the fusion, after the fertilization, we say that it is diploid. Now, there is chance that there is a 50-50% chance that the zygote can be male or female. If it has two X chromosome, then it will become the female zygote. And if this gamete has fused with this gamete or either of this gamete, now we will get a male zygote having one X and one Y chromosome. The zygote, it divides and ultimately gives us the embryo. So embryo is a mass of cells. So it is an undifferentiated mass of cells. Now the embryo, it starts to develop and entering into first trimester, second trimester, third trimester, it develops into fetus, which is the unborn baby, unborn infant or baby inside the uterus. Now, this is this was the process of fertilization. How the gametes, the gamete formation takes place because of the process of meiosis. And after meiosis, the gamete formation takes place. The gametes, they fuse together. And this process is called fertilization. And after fertilization, we get the zygote, which zygote is a single cell which is diploid. The gametes are haploid or monoploid, and the zygote is diploid. This diploid zygote then undergoes the process of mitosis. The zygote divides the cell division, the process of cell division that is taking place here is the mitosis, and this mitosis is given rise to embryo, and then embryo, which is an undifferentiated mass of cells, that ultimately gives us the fetus or the unborn baby. Now, there's a concept of twins. Let's discuss the concept of twins. So we have two types of twins. We have identical twins and we have the non-identical twins. You must have seen in your in your neighborhood or in your family, you must have seen the cases of the women or the parents having a twin babies. Okay, so either the twins, they can be identical or they can be non-identical. Most of them are non-identical. There are just the identical twins are somewhat rare. So these non-identical twins are called the dizygotic twins. And what the term dizygotic is referring here is that it refers to that the babies or the twins are coming from two zygotes. So the two zygotes are forming. That is why the name dizygotic twins. They are also called the fraternal twins. Non-identical, dizygotic or fraternal twins, one and the same thing. So here we have, in female, we have two ovum or two eggs. They fuse and fertilize with two sperms and we get two zygotes. And then these two zygotes, they develop into two individual babies. Now, a normal human female, typically in 1 month, her ovaries release a single egg or a single developed ovum. But sometimes, like multiple ovums also can be developed, and a male can produce 100 million to 500 million sperms at a point. So it is, if more than two eggs are there, so it is very much possible that the sperms will fertilize that. Now, the possible combination of babies that we can get from this non-identical or the dizygotic twins is that one of the combination is that both the babies can be male. Then we can have one male baby, one female baby. And we can also have both the female babies because two different sperms are fertilizing that two different ovums. So ovum, we have this X chromosome, but the sperm that is fusing with that particular X chromosome might have the X chromosome or might have the Y chromosome. So depending on which of the sperm containing either X or Y chromosome is fusing with the ovum or the egg, then we determine the sex of the child. Okay. Now, the dizygotic twins or the non-identical twins are just like any other siblings, but the only difference is that they are born at the same time. Okay. Now we have the monozygotic twins or the identical twins. Now, identical twins or the monozygotic twins. So monozygotic, what does it infer that now the babies or the identical twins that we are getting are from the same zygote. Okay, they have originated from the same zygote. So we have one egg or one ovum from female fertilizing with one sperm, we get one zygote. Now, because of some splitting or because of some random reasons, uh, reasons we do not know, it is under research. Due to some random reasons or random circumstances, this zygote it gets split and it makes two different embryos. Okay, so in the dizygotic twins, we had two sperms, two eggs, giving us two zygote that ultimately is giving us two embryos and two babies. But now we have one egg, one sperm giving us one zygote. This one zygote is giving, due to some reasons, two, because of some division, it is giving us two embryos. And these two embryos now develop into two different babies. Now, what is the possible combination here? The possible combination here will be either there will be both males or they will be both females because only a single sperm and single ovum are involved. Okay, so if this is X chromosome of the egg and the Y chromosome of the sperm, it has fused, then in the zygote we have this XY combination. So the child will be a male child. And now because of some reason, this zygote is giving us two embryos, then both of the embryos will develop into the male child. Similarly, if the X chromosome is there in sperm, then we will have two X chromosomes in the zygote. And if the zygote splits and giving us two embryos, then these embryos will develop into two female child. So, and these will be identical. They have the identical same, they have the same genetic makeup. Genetically, the identical twins are the same. There is some variation is that a special term is given to Siamese twins or the conjoined twins. So you must have seen in some news reports or somewhere that some of the twins they are born attached. Either their heads are attached or their back is attached or some part of their body is attached with the other part. This happens because there is no proper uh, separation of the two embryos or the zygote. So zygote is not effectively it is getting separated. Okay. So zygote it has split into two parts into giving us two zygotes. But ultimately what is happening is that they a part of the embryo it remains attached. The part of the embryos remains attached. So ultimately the fetus they remains attached and the babies that come out are attached. And they please note that they will always be identical. So conjoined twins or fraternal twins are the twins that are born attached and they are always identical. Now, while we were discussing about this genetic matching, so what is genetic matching? So an individual. So suppose you and I, we are individuals. So An individual's genetic matching with his or her parents. What do you think that genetic matching of me with my mother will be, or me with my father will be, or your mother and you or your father? So, an individual's genetic matching with their parents is 50%. It is because you have got 50% chromosomes. Remember, you have got 22 plus X from mother. You have got 23 number of chromosomes from mother and 22 plus X or 22 plus Y from your father. So you got 23 chromosomes or half the number of chromosomes from mother and half the number of chromosomes from the father. So that is why any mother child or father child or like genetic matching or with the parents is 50% for any individual. Now, an individual's genetic matching with his or her sibling. So, your genetic matching with your brother or your sister, what do you think it will be? Will it be more than 50% or will it be less than 50%? So, it will be 75%. It is greater than 50% because you have the same set of parents. But it is less than 100% because of a phenomena known as the recombination. Do not have to go in much detail about this recombination. Just remember, so this recombination is a phenomena that is preventing the siblings from having the 100% genetic matching. Now, these siblings, they are the preferable donors during the organ transplantation. So, if organ transplant, so suppose if a person X needs a organ transplantation, so the best donor there will be, yeah, as compared to the mother, father, or the sibling will be the sibling is the preferable donor. The best donor will be the identical twin if that other person is having. So identical twin because they have the same or the 100% genetic matchup. Okay. So genetic matching is 100% in case of the identical twins. So they are the excellent or the most preferred donors during the organ transplantation. But not everyone has their identical twins. Identical twins are very rare. So the second more preferable will be the siblings as compared to parents. Because with siblings, we share the 75% of genetic matching as compared to 50% with the parents. Now, what do you think the genetic matching with our grandparents will be? So, an individual's genetic matching with his or her grandparents will be around 25%. Okay? Please make note of it. Now, we discuss the stem cell technology. Like we have been discussing about the organ transplantation. So, when we are discussing about the organ transplantation, then the stem cell technology is needed. So stem cells are the cells that are converted into the other cell types. So we have different cell types in the body. So we know that it is the from the cell theory that a cell arises from a pre-existing cell. Yes. So we know that a cell arises from a pre-existing cell and the cell divides by the process of mitosis or in the case of gametes by the process of meiosis. Now, um, and we know that there are two types of cells in the body: the sex cells, that is, that are present in our gonads, and the somatic cells, the cells that are not the sex cells. So, skin cell will divide, okay, and will be convert, it will give rise to the skin cell only. A liver cell will give rise to more and more liver cell. Okay. Muscle cell will give rise to more and more muscle cells. So the type of cell is not changing. Okay. One type of cell is giving the same type of cell. Now, in case of stem cells, the difference is that these can be converted into the different cell types. So now we have a cell, we can make different type of cells. So we can get the skin cell out of it, we can get the bone marrow out of it, we can get the blood cells out of it, we can get the liver cells out of it. So that particular cell that can be converted into different cell types, which is giving the different cell types, that is the stem cell. Now, most adult somatic cells are not stem cells. When we grow, when we become adults, so the somatic cells in our body, somatic cells like your skin cells, your liver cells, your kidney cells, your muscle cells, and these are the somatic cells, your nerve cells. So these are the somatic cells that are not involved in sexual reproduction. So which are not the sex cells. So these adult somatic cells, they are not stem cells. The application of this stem cell technology or this stem cell concept is that the stem cell technology aims at making copies of one's own organs for transplantation. So when we do this organ transplantation, our immune system, there is high chance that our own immune system might reject the implanted or the transplanted organ. Okay, because our immune system, it has a very strong sense of self and anti-self. What it perceives as self and what it perceives as anti-self. So self is tolerable, and something that is foreign, that the immune system considers as anti-self, it tends to destroy that. So in transplantation, it becomes very important for us either to suppress the immunity, but if we suppress the immunity, if we suppress the immune system, then we might develop some secondary infections. And if we get the organ transplanted from some other source, from a foreign source or foreign donor, then there's chance that that particular organ might be rejected in the body. So the best way that will be if we can make the copies of the organs from our own stem cells. So these, they will be self, and our immune system will not react against it or will not reject it. Now we have different types of stem cells. So stem cells, depending on their potency, depending on like how many number of cells, how many different type of cells they can divide or they can be converted, these are based on their potency. They are the totipotent stem cells, they are the pluripotent stem cells, multipotent stem cells, and oligopotent. So from totipotent to pluripotent, the potency is decreasing. So totipotent are the cells, are the stem cells that can be converted into all the other type of cells in the body. All the body. So totipotent stem cells can be converted to into all the other cell types. And what are the other cell types? That is the body cells, the umbilical cord cells, and the placenta. That is, and the example, the most important example of a totipotent stem cell. So totipotent stem cell is a cell that can be converted into all the other cell types. Other cell types, that is all the cells of the body, the cells of the placenta. So placenta is a tissue that connects the developing fetus with the mother, and the umbilical cord. Okay. So umbilical cord and placenta cells, like they are responsible for the healthy development of fetus. So fetus, it takes its nutrition from the umbilical cord and the placenta. And placenta also makes sure that the mother and child blood is not mixed during the pregnancy. So the placenta cells, the umbilical cord cells, and all the cells of the body. So totipotent stem cells have the ability that they can be converted into all the cell types. So the keyword here is all. Example being the zygote. We know that zygote is a single cell which is diploid and all the cells of the body. So a single zygote is ultimately giving rise to an entire organism, like entire human being. If it is giving rise to the entire human being, so that means that particular zygote is a cell, and that zygotic cell, has the ability to be get converted into any of the or all of the other cell types. That is our body cells during our fetal stage. That is, it has become the placenta, and it has also given rise to the umbilical cord cells. The pluripotent stem cells, on the other hand, are the cells that can be converted into almost all other cell types. So, totipotent can be converted into all the cell types. Pluripotent can be converted to almost all the cell types. That is, it can be converted into all the body cells. The example being the embryonic cells. So the embryonic cells that we have, okay, they can be converted into all the cells of our body, except for the placenta and the umbilical cord cells. So totipotent stem cells can be converted to the body cells, the umbilical cord cells, and the placenta cells. But the pluripotent stem cells can be converted to all the body cells, not the placenta cells, and not the umbilical cord cells. So we say that they can be converted into almost all the other cell types. Example being the embryonic cells. Then we have the multipotent stem cells. The multipotent stem cells can be converted to many other cell types. So the potency is decreasing. Totipotent all, pluripotent almost all, multipotent many. So they can be converted, the multipotent stem cells can be converted to many other different cell types. Now, example here being the umbilical cord cells. The umbilical cord that attaches or that connects or that is important for the developing fetus as it is connecting it with the mother. So, nowadays, the stem cell banks, they are preserving this umbilical cord of a newborn child. It is because the cells of that umbilical cord, they are multipotent stem cells and they have the ability to convert into many different cell types of the body. So if in future the child or the baby, if he or she may need certain organs for transplantation, then his or her stored umbilical cord stem cells can be used to make the uh, copy of the organ that needs to be transplanted. So it is a awareness is less. There is less awareness, but it is a very good practice to store the a baby's umbilical cord stem cells in the stem cell banks. Then we have lastly the oligopotent stem cells. They can be converted into few other cell types. So we see that the potency or the potential to give uh, different type of cells is decreasing from totipotent, pluripotent are lesser potent, multipotent are even lesser potent, and the oligopotent they can only be converted into a few other cell types. Example here being the bone marrow cells. So the bone marrow cells are the cells that can be converted into the different type of blood cells. So we know that the cells of the blood are produced in bone marrow. So the cells of bone marrow, they are the stem cells and can be converted into the different cell types. Okay. So it is only limited to a few cell types. That is why we call it the oligopotent stem cells. Now we discuss about the induced pluripotent stem cells. So we know that the somatic cells in the adult body, the somatic cells, they are not naturally the stem cells. So we induce them to become the pluripotent cells. Pluripotent means they can be converted into different types of almost all the other type of cells. And we know that the embryo is the pluripotent stem cell. So what we are doing is basically we are inducing the somatic cell to behave as the to behave as the embryo cell and become a pluripotent cell. And how we are achieving it? We are achieving it by activating some embryonic genes in the somatic cell. So in the somatic cell, there will be certain genes that were active during the embryonic phase, but as the child grows or when the embryo is developed into a fetus, these genes they get inactivated, and they get inactivated so that particular somatic cell is not working as a pluripotent stem cell. Now, if we activate these genes that are present, and if we that are present in the somatic cell, these inactivated genes, if we activate them, then this somatic cell starts to behave as a embryonic cell, and embryonic cell is a pluripotent cell. So this was a remarkable and groundbreaking discovery on how we can convert the somatic cells into the pluripotent stem cells. And the Nobel Prize of 2012 was awarded on this research, and it was given to Shinya Yamanaka of Japan. So that was our discussion on the stem cell technology, and we have discussed about the genes and the genetic matching, the mutations. Let's end this class today. In the next class, we'll be discussing about the other aspects of biotechnology. We'll be discussing about the genetic diseases. We'll be discussing about the biotechnology techniques and what are their applications. We'll also be learning about a bit about the Human Genome Project and the fertility techniques that can be used by the couples or that are used globally. So, thank you.